So obtaining a Nobel Prize is a fantastic honor. It's the best you can obtain as a scientist. And just to start this out, this is what it looks like, right? I'm very honored that the Nobel Committee has actually pointed towards click chemistry for this prize. And the reason is I think click chemistry can solve a lot of the problems that we are facing out in the future. It makes us able to do molecular architectures that has never been possible to do before. Already back in my early days, I know very well both Barry and Caroline. Caroline, I discussed a lot of chemistry with her when she was a graduating PhD student back in the early 90s. And then Barry, I visited several times, and in particular, I visited him right after his first Nobel Prize. He is one of the few who got 2 Nobel Prizes. So they are very good friends of mine, but we have never worked together. All the discoveries of click chemistry was done independently in the 3 laboratories. So when you get a Nobel Prize, you also get the right to come up with some mantra about what you think is right and wrong. And this is a very nice situation. You are able to actually influence politics in your own country and maybe even abroad. So here is my mantra. First of all, chemistry is everything. Without offending anybody, chemistry also includes biology, nanoscience, and so on. All of those are molecules that interact with each other, and when it is that, it is chemistry. When we breathe, it's chemistry. When we feel pain, it's chemistry. When we are sleepy, it's chemistry. When we feel hungry, it's hormones that goes from the stomach to the brain. So if chemistry is everything, why don't we teach our kids from first grade about chemistry? We teach them how to read and how to write and math. It's a little bit old-fashioned only to have these 3 subjects from first grade. We should teach them how chemistry works, not by formula, not by all this difficulty that causes chemistry to be the evil science, but rather by visualizing this chemistry, putting it into the brain of the young people very early on. And that makes kids love it and play with it, and that's what we should continue to do all our lives. So research without no constraints is the next issue. Can we actually be allowed to do the research that give us the new big breakthroughs? So of course we should have applied research. Of course we should solve the problems around us. But we should also have the ability to follow whatever is there that is very exciting. So funding by performance could be a better way to fund than actually making an application which contains CO2, sustainability, genes, and so on. Rather than that, we would make research based on our observations in the laboratory. So I started out very early getting an interest for science actually all the way back to my childhood, because science is what makes nature work so fantastically. So if we want to unravel what happens in nature, our own lives, we need to understand the chemistry. And this is how I started at Carlsberg Laboratory, later on developing a platform that could investigate all of that. So we have combinatorial chemistry. I was involved very early on in combinatorial chemistry here, and we can use assays that are based on the polymers that we make, getting active compounds that are then assayed in different biological systems. We can work with the peptides or small molecules like these ones, or even catalysts like these ones. We can add properties to our polymers that allow us here to grow a monolayer of cells as a detector of activity of the compounds that are on the beads, and that's what you see over here. And that brings us up to this one. How do we then decipher the molecular structure in the beads? Well, we use a barcode. Each of these small beads, each of these small polymer particles contains a barcode that we can read at any point. And it's small microparticles that are immobilized in a three-dimensional matrix. So at any point of the synthesis, any point of the assaying, we just read this code and we have the structure of the molecule. All of these things provided me with a platform for combinatorial chemistry. And that is what we were doing here, showing our instrumentation, which is a very simple multi-column synthesizer, where we can easily mix all these beads and put them back into columns again and increase exponentially the number of compounds with the number of cycles here. So in a week, we can make 1 million different compounds and assay them. We can add an enzyme, for example. So we add this enzyme to the beads. It goes into the interior of the polymer, and we see some of the beads will light up because of a fret substrate that is attached to that bead. And we can take it out and find out what was the activity of the enzyme that we added. So this is sort of the basis of our technology. The other basis is, as I said, the polymers. We have particularly 2 polymers, the Spock resins, which is an all-ether resin, very good for carbon ion and carbonium ion chemistry, and the peptide-like resins, which is also PEG-based, and that is the Pica resin. Is compatible with both organic synthesis and with biology, so it makes a unique platform for combinatorial synthesis and iterative selection. Combinatorial chemistry was very game changer at that time. So what we wanted to do was to take all the organic reactions and optimize them so they get just as efficient as the peptide and DNA chemistry that is carried out on solid support. And here you see all the chemistries that we went through. Disorder up here at the top, aldo, nitroaldo, Wittig, redox reactions, and so on. A lot of reactions can be carried out, and not only organic reactions, also aqueous reactions actually can be carried out on this kind of resin, because the resin is compatible with all the spectrum of solvents from toluene all the way to enzyme buffers. Not only could we do organic reactions, but because the swelling of this resin was like 20-fold, we could have proteins like all of these proteins over here. All of these proteins could be transported into the polymer and carry out reactions, enzymatic reactions on solid support. So we had a whole technology platform that would work for anything we would like to do. And we were working for a beer company, so we even immobilized yeast cells in this support and used them for fermentation. Yes, we could actually make these supports in a very controlled manner, getting almost evenly sized polymer beads that swells up in anything. So during this work, we were working, as I said, with the disorder reaction. And one of my PhD students, Christian Tornø, who is down here. Yeah, this is Christian. Christian was working on this disorder reaction, trying to place a triple bond in the middle of a peptide for doing disorder chemistry, branch out, make new Novo Nordisk drugs. But that didn't succeed at all. So after a couple of months of working on this, Christian came to my office and said he would much rather work at Novo Nordisk because this didn't work. This was a crappy project. So I asked him what was wrong, and we looked into his data, and I noticed the proton here, this proton here from the triazole in his products was always there. The product was very clean. So what was it? It was this triazole, and he then started to investigate the compatibility of this reaction with all the different protein functionalities and carbohydrate functionalities. And it appeared that it was completely compatible with everything. So that allowed us to do a whole new set of things. And what is it really that makes it so selective? So this is an alkyne. And here you see the the HOMO of the alkyne, the pi bond, one of the two pi bonds, and the electrons there are asking for a positive charge. They are nucleophilic electrons in the pi bond. So they are asking for a positive charge, and it comes here, it's a copper-1 ion. Many other ions have been tested, and copper-1 has a very specific interaction with triple bonds. I'll show you a little bit later. So this here, it acidifies the proton on the triple bond because it extracts the electron density, and then, well, nothing happens. It just looks like that. So now we have made sort of the standard nucleophile of an alkyne using copper-1. That's not enough. Nothing happens with that one. We need a second copper atom, so it comes in here. It attaches itself to the pi cloud, and it strips off the electrons from the triple bond to form two equally positioned copper atoms sitting out here at the end of the alkyne. And now we have transformed the alkyne from a nucleophile into a very good electrophile. So this is the LUMO of the alkyne. The empty orbitals of the alkyne is looking around for electron density, and fortunately, here comes the A site. And the A site fits exactly with the phases and the overlap and the sizes and everything. And that is the key to the success of the click reaction. So here we form by a ring, what is it called, a pericyclic reaction. We form a heterocycle with a copper atom inside the ring. It loses that one and forms a triazole. It loses a second one, and here you have the triazole product, which gains a lot of energy from the aromaticity, and that's a driving force for that reaction. So is that exciting? Not very exciting, because we just connected a phenyl ring with a methyl group. So that's not very exciting in itself. But the fact is that if we look at this reaction, it can actually do very complex things. In the literature, we will find, on the crystal databases, we will find all these copper alkyne complexes that illustrate every single step up here in this cycle. So if this one would work, here it is. We start out up here with coordinating the copper to the alkyne. We get the coordination of the A side, and we get the attack of the A side nucleophile on the electrophilic end of the alkyne copper complex. Then we get the extrusion of one copper, and the other copper can be replaced with an electrophile. And then we are back to the starting point again. So two things. One is that we have all the different complexes. Here is one with copper at the end of the alkyne and one sitting on the alkyne itself. Here's one with two terminal copper atoms. And here's one where we have two copper atoms attached to the triazole product. So all these variations is there. There's probably hundreds of these kinds of triazole copper complexes. Another thing that one notices is that if we actually make a ligand that can bind two copper atoms in the right orientation for making this kind of complex up here, we get an incredible increase in rate of reaction. So if, for example, we look at ethyl propionate with 9 molar percent acetic acid, and compare that without the catalyst, just this reaction. This is the orange one, and when we add this catalyst, without the catalyst, we get this one just with the copper acetate, you get the grade one. And so the catalysis is very, very strong when we have the right orientation of the copper atoms. So what can we do? Why is it that we get this Nobel Prize out of this? The reason is that we can, for the first time in history, really make molecular robots which takes a functionality that we have isolated from nature, another functionality, and combine these two to make a molecular robot. So here are the three guys we want to combine. There's one here, and one here, and one here. The first one can kill a cell by interfering with the programmed cell death. The second one, it can locate tissue, say a cancer cell. And the third one can import into the cell. It's a cell-penetrating peptide that we're currently working with. If we now substitute these with using amper stop codon technology or just synthesis, substitute this with alkynes and acids and mix them into a container containing the copper-1. And here we need to completely avoid any kind of oxygen. Then we get the molecule formed 1-to-1, and we can isolate that in high quality and use it for biochemistry. And this is the reason, the argument that they used for defining that we could do something new with click chemistry. So is there no problems associated with all of this? Well, Sharpless noticed that the copper catalysis didn't really work when there was oxygen present because the copper-1 was oxidized to copper-2, which is completely inactive in this reaction. So how do you then maintain the copper-1? Well, you can protect it, or you can add a reductant like this one here, ascorbate. That can take copper-2 to copper-1, and then we have an active catalyst again. Aber that's not a good way to do it, because in this process, we are generating a very large number of reactive oxygen species, radical anions, radicals, and so on. And these are very harmful for biomolecules. So whether this would work very well if we take phenylacetylene and couple it with an acide, an organic acide, it won't work with biomolecules where you have oxidation of all sorts of side chains in the protein. So we have to be aware that this happens and make free thaw cycles and bubbling and all of that in order to make this oxygen-free conditions, and then it works like a dream. So I'll give you some examples of how we have used this. This is our first real evidence that you could use this for disulfide bond mimetics. So take proteins and replace disulfide bonds, which are, of course, possible to reduce in the biological system, and replace them with something that is completely stable. So this is a Japanese horseshoe crab. It looks like a tank, and it actually works like a tank. It has lived for 400 million years. Essentially, not this one, but they have been here for 400 million years, and they have a very unique innate immune system. We actually harvest this. There's factories where we take them and milk their blood and use it for detection of bacteria in blood. So it has two macromolecules that engage the foreign organism, and then it injects these kind of compounds up here. These ones here called targetplacines. Targetplacines are antibacterials that can kill. So we were very interested in looking at these antibacterial compounds to see, can we make something which is not subject to resistance in bacteria. And this is what the targetplacine looks like over here. And you can see it's a peptide sequence in one letter code with two disulfide bonds, and we replace those with different lengths of click bridges. And we still get this ribbon-like structure. We investigated this structurally by NMR and could see that we have NOEs here that indicate we have a dimer in solution. So this molecule is swimming around like a benign cannonball, positive charge on the surface. It then finds a negatively charged bacterial surface, and attached to that, it will find the membrane, and then it will open up and form a pore in the membrane, and all the contents of the cell will come out, and the cell will die. So that's how it works. Now, is that active? Is this process actually working in all the different cells that we want to kill? Well, for these 4 organisms that we tested out, so coli, Staphylococcus epidemus from hospitals, Salmonella typhimurium, and Bacillus subtilis, we tried these 4 organisms, and we found that targeplacin was super active in most cases, but there's one case here where it's completely inactive. And this inactivity is probably a result of disulfide bond reduction. So we asked ourselves, is our compounds as active? And they were actually just as active as a natural one, including for Bacillus subtilis. So this means that we can actually use this kind of bridges to substitute for disulfide bonds and get bioactive molecules. Interesting observation. Here's another example, an example where we are looking at the expression of proteins. Proteins is a very, in particular proteases, is very difficult to express. While an antibody can be expressed at multi-hundred milligrams amounts in 1 liter of fermentation broth, we cannot do that with this proteases, because the protease is a toxic compound. It will hydrolyze the essential proteins inside the host cell. So therefore, as soon as we start getting folded protein in the cell dies. And what can we do about that then? So we asked first if we could do it in the coli, and they weren't very happy with that at all. So we then decided to cut the protein in 2 and make them separately using the amber stop codon technology. So we are taking 2 containers expressing the 2 halves of the protein, and we are getting hundreds of milligrams of each of the 2 parts. So far, so good. Using amber stop codon technology, we could actually include these residues which allow us to cyclize or to connect the 2 halves with each other and see if we could get a folded protease. So we asked the coli if that is okay, and it was very happy with the result. This is showing a more scientific scheme of the same thing. We used 2 different bridges, a longer bridge and a shorter bridge here, installed them via the amber stop codon technology. They would form the shorter bridge or the longer bridge shown here. And the shorter bridge very short, the shorter bridge didn't work totally. It gave us a lot of unclicked material with the optimized conditions, while this one went 100% to the product. So we mix them 1-to-1 and add the copper 1, and we get 100% of active protease, we hope. One of the things that we observed is that we could produce these very cleanly, in particular when we have the histidine tag placed after the stop codon. Very important observation. It was much more difficult if the his tag was on the upstream side of the stop codon. But we got all of the compounds that we wanted to have in fairly good yields. So this is the HPLC, and this is the mass spectrometry of a fragment. This is the mass spectrometry as well. So if we look at then the connectivity between the 2, we have the short linker and the long linker here. This is the one with the short linker, and this is the one, or is it the other way around? This is the one with the long linker, sorry. This should be the one with the, no, that's correct. It's because we have left out some of the residues in this longer linker. So here we have the short linker, here we have the long linker. And what we see here is what happens if we don't remove the oxygen from these 3 reactions. You can see there's 16 between all of these peaks, and that's simply oxidation of the side chains. So we probably have something like 10% yield in this reaction. Whereas if we remove the oxygen completely and do the click reaction, we get a very nice product. So we have these two fragments, and can we then combine these and get an active protease out of it? Here's the two fragments, and there comes the copper, and we form a triazole. And we ask the question, is that active? And this is by cleaving a substrate shown up here. So this substrate here can cleave at that position, and we can see by the thread whether we have activity, and you can see our protease is a little bit better than the natural one. So that's very nice and comfortable. And I think this could be a method to generally make these kind of proteolytic enzymes by expression. So here we are in Stockholm. Very nice week. I can highly recommend it. It's 1 week of Alice in Wonderland, and we have a great experience. It starts out by you being collected in the airport, not by the entrance of the airport, but at the airplane. So they go out to the plane and get you in a limousine, and you are driven into Stockholm and Cotisches and all of that. Fantastic. And here we are at the Nobel Museum, getting informed about what is going to happen during the week. You deliver a small item that meant a lot to your research. And this is now at the museum of the Nobel Museum. This is the world's first multi-column synthesizer made in the neighbor's garage. And he had a big lathe, and I was allowed to use that to make this instrument. It has 20 columns, and you could make 20 peptides in parallel. Or you could make combinatorial libraries by doing different couplings, out with the resin, in with the resin, another coupling. So that was the starting point for our combinatorial chemistry as well. So that's up there together with the publication that we describe it in. And then you are signing a chair. I'm very proud here because my name is right next to Emmanuelle Charpentier. I don't know if you know her, but she is a lady of great power. Then I go on to the next subject, which is proteins. Proteins, not as they are made by nature, but as we would like to make them. So this is a protein from nature, interleukin-4 receptor and the papain here. And what you notice on this slide is that I have indicated the hydrophobic core. In my opinion, the hydrophobic cores are one of the most important properties in proteins in the folding process and in the activity of those proteins. So these hydrophobic cores are extremely difficult to make. You learn that by trying to make them for 40 years. It's very difficult to make the fitting of the hydrophobic residue so you have no pockets for water inside in the interior of the protein. And also to have the right mobility of everything on the outside controlled by these hydrophobic core interactions. So hydrophobic cores are essential. And we would like to make proteins, particularly we would like to make them by synthesis. So proteins are very large and they have secondary structures. They have a beta sheet that goes here, another alpha helix that goes there, a beta strand that goes over there, and another alpha helix. And they have to come together in this folding, creating the hydrophobic core and the active site residues. So the active site is tiny compared to the size of the protein. Maybe there's something, a cost-benefit analysis of the protein structure shows you that it's very difficult to make something like that with only 20 amino acids. So if we had many more amino acids, maybe we could make something like that much smaller, still have that activity, because the tacticity of the amino acids, the L amino acid use, makes it tactic just like a polymer, having crystallinic structures like beta sheets or alpha helices or so on inside the molecules. So we would like to do that with a mixture of L and D amino acids. All the 5,000 L and D amino acids that are available out there can be used, and then we would do it by synthesis. So we start out by modeling. We model a fold, a random L and D amino acid sequence. We let it fold up. We look at the core and see if we do it many times. We choose a fold, that often happens, and then we optimize that fold by inserting amino acids mutationally into the core until we have a perfect packing and a perfect surface for modification with hydrophilic amino acids. Then we look at the protein and see is there a good site that we can use, a cleft, for example, that we can use as an enzyme cavity. And we insert the active site residues, and whoopty, we get an active protease. So that several different ways we can come from the unfolded structure to the actual product, either by modeling on a surface of a protein or modeling separate out in the open. So this is what we do. We call it mutational molecular dynamics, in which you mutate, do a short dynamics, mutate, do a short dynamics, mutate, do a short dynamics, and then we very rapidly funnel into a low energy situation. And we continue to do this until we are completely happy with the fold and the surface and the solubility and the shape. Once that is done, using this cycle, we will then introduce the click bridges. We will introduce the active residues, and then we will see if we have an active protease. And here are 4 such proteolytic enzymes, very small, synthetically tractable, and they are, some of them, active. I will focus today on this one up here, which is a serine protease with serine, a histidine, and a carbonyl group that interact there. Now, these foldings are very stable. This is several nanoseconds, 10 nanoseconds of calculation. Nothing happens to the structure. It's completely stable. And it's stable before we do the click bridges. So the click bridges are just adding to that stability. It's Mads, Rosen, Sarah, Line, and David who is working on this kind of projects. So this serine protease, it is small like that. It's very stable on its own. And when we introduce the click bridges, so you can see one click bridge up here on the top and a disulfide bond here. So we use both in this case. And we get a very nice orientation and very high stability of the active site, as you can see here. This is less than 5 in RMDS. So very nice stability. We can also synthesize it. This is how clean it is off the synthesizer. Very nice. And this is the MS of the right protease. So making things synthetically tractable means that now we have the world in front of us. There's nothing we can't do. We tried it out on a library of substrates. So this is a FRET library substrate. And we have here 3.2 million substrates, so we can actually try the protease out, see what it does. It cleaves, and when it cleaves, it gives us a fluorescent bead. So we tested this out with subtilisin BNP, shown here, one of the fastest proteases in the world. And this is what we saw. You can see after 5 minutes, nothing has happened. Here, after 10 minutes, we start to see a small ring around this bead. Over here, the ring is growing. We get more and more activity. After 24 hours, a lot of the beads are giving activity. But you can see it's highly specific initially because it's just this one that gets a ring. So we tried with our protease, and this is the result. You can see after 2 hours, we start seeing some fluorescence. After 3 hours, we have quite a lot of fluorescence. Here we see after 19 hours, 24 hours, 7 days. And just to make absolutely sure we have good proteolysis, we have left it for 90 days. And then you see clearly that all of the substrates has hydrolyzed at this point. But initially, we have a lot of specificity in our hydrolysis. So we have made a specific proteolytic enzyme. So what does it constitute? This is a compound. It has the two bridges. One is a click bridge, the other is a disulfide, and we can make those individually. We have a serine, a histidine, and a aspartic carbonyl group that makes the active site. We have a hydrophobic core. It's fully soluble because of the surface is polar. So it's fully soluble, 4 milligrams per milliliter, for example, which is quite unique for protein. It has this sequence, which is essentially gibberish for most people, because it's not the normal one-letter code. There's all sorts of things in there. And here's the surface. You can see the polarity of the surface is quite good, allowing us to have soluble protein. So we are back in Stockholm, and this is a ceremonial dinner, a very nice day where we go to the city hall, 1,300 people. You get the medal first. And this is the king delivering the medal. So the king of Sweden comes with the medal, and he takes your hand, give you the medal, and then you stand a little bit like that. And he doesn't really release it. So you try to take it out of his hand, but he actually takes it and goes back to his seat. So you wonder what is happening, but I was taught later on that this has to do with the fact that a lot of the Nobel Prizes are given, like in my case, 20, 30 years after the discovery. So some of the Nobel laureates, so they forget their Nobel Prizes under the seat in the ceremonial hall. And you actually get the medal at the end of, where did I put it? Somebody took it. There. You get it at the end. So this is the Nobel Prize you get the last day. So I would like to now turn to structural integrity by clicking. We can make molecules that are very highly stable because of the click bridges that we can make in these molecules. So maybe we can actually take something like a small molecule and make it high affinity, just like antibodies. So instead of 160 kD, we can operate with 3 kD. That would be very advantageous. So we are making these kind of beta hairpin molecules that can bind to protein surfaces. They are structured in this way. They are threonine rich. So on one side of the beta hairpin, you have threonines maintaining the beta structure. On the other side, you have recognition residues that can recognize the surface. And then you have a turn and a bridge in the end. So they can actually be made for the first time as two small molecules recognizing each other with a high affinity. So this is two molecules that recognize each other. The problem is really in the modeling. You have to be very aware that the modeling of intermolecular interactions in force fields is actually wrong, because it looks at the positive charge at the nuclei of the atoms instead of the electrons that meet each other in the surfaces. So for me, it would be much better if we had a software that could simulate all these small dipoles in between the two surfaces. But we are able to get there eventually by optimizing this, looking at it visually, and so on. So here you see basically a negatively charged surface and a positively charged surface, not in terms of charges, but in terms of electron density, interacting with each other and forming a good sign. We have seen this already, so I will not talk about that. But here is a binding study directly on one of our resin beads, and you can see the binding constant is approximately, is approximately here. So it's about 700 nanomolar for two small molecules recognizing each other. That's quite nice. But we can go much further than that by looking at protein interaction. So this is green fluorescent protein and interleukin-4 labeled with ROX. So one is green and the other one is red. Now we have made the two beta bodies, one which recognize green fluorescent protein, one which recognize interleukin. So here you have the green fluorescent protein beta body. Here we have beta body for interleukin-1. If we now add green fluorescent protein, only one of the beads goes green. If we add the ROX, only the other bead go red. So there's clearly selectivity, and we have quite good affinity as well. Now, if we click these together, we increase the affinity significantly of these molecules. So here we have ROX on interleukin-1 beta and ROX on interleukin-1 alpha. Very similar protein folds, just a few amino acid residues that are different. And you can see the recognition is only towards one of them. So just as specific as an antibody recognition. And the other thing is this increasing strength can actually be used. So here we have a sandwich assay in which we have made one of these beta bodies towards interleukin-6 on the bead, and the other one towards interleukin-6 in another site on the protein with a fluorophore. When we mix these two beta bodies, one on the bead and one in solution, nothing happens. Then we add 20 nanomolar of the protein, and miraculously, we see a very red bead because the protein is attracted to the one that is on the bead, and the other one is attracted to the protein that is now bound to the bead. So we can make a sandwich assay showing very high affinity towards this protein. With that, I would like to now go to our results generated within the field of Alzheimer. Alzheimer is a problem that is going to concern all of us. And almost everybody has somebody in the family who is suffering from dementia because of Alzheimer. This is a disease that is caused by plaque formation in the brain and neurotoxicity around these plaques. So if we take the Alzheimer plaque and we look for particular structures, we will find that this structure here is present in all the plaques, and it has a crystal structure shown here, composed of two long fibrils sitting together. This is the main crystal structure in fibrils. There's a lot of other crystal structures, and it depends on whether we have Alzheimer, a beta 1 to 42 or 1 to 40, what kind of crystal we get. But now we would like to know, why do we actually get neuronal cell death if we have oxidative stress? It should work, and it doesn't. I need to go there. Something is wrong in our picture of why the cells are dying. One of my master students, Nicholas, tried to take this fibril and look at the surface in the computer, surface for the growth, where the fibril grows, and see if we can make a beta body that sits on that. We designed this one. And we designed actually a whole range of them. If we made a beta body to the outside, it actually increased the fibrillation quite significantly. If we made them to the ends of the fibril, we could reduce the fibrillation very significantly. And this is shown here, the full fibrillation by a fluorescence assay, where this one, shown here at the top, is straightened out when it inserts into the fibrillar nature of the fibril. And we can actually break the starting fibrillation. We can break that by adding the peptides. Some peptides are more efficient than others. And you can see some of them can keep it almost completely from fibrillating. But this was at 25 degrees. When we go to the human brain temperature of 37 degrees, there's almost immediate fibrillation of these peptides. And you can see in the presence of peptide, we get a little slower fibrillation, but it still happens. So our idea was maybe we should make one that sits on both ends of the fibril, because of course there's a different structure on the two ends of the growing fibril. We did that, and now we could completely abolish fibrillation here. So this is the normal fibrillation curve here. Over here we have a fibrillation curve after addition of these two peptides. Works quite well. No. Usually, this is moving. This is now moving. So now we are just need to get out of the presentation. And then in again. No, I know what you should do. I know actually. And now we can go in with this. Sorry about that. Oops. Yes, here we are. Sorry. When we did the fibrillation first and then add the peptide later on, we found here after 250,000 seconds, so it's a long experiment, we added this peptide, and we could see that we could actually remove some of the fibrillation, but not all of it. And that is most likely because in this fibrillation, we have formation of cross-linking between the fibers. This is a well-known phenomenon. So we can remove everything that is made recently, but it's very difficult to remove the long-term fibrils. But we also thought maybe we can improve this by making a hydrodynamic shell on the one side of the beta body, so that on the structural side we would have a large water shell that would improve the interaction with the fibrils. So we inserted glutaminic acid. This is glutaminic acid here. And we just needed to protect the hydroxyls and put on an FMOC group, and then we could use that as a building block. This took a whole half year to make that building block. It was not easy, but it succeeded in the end. And we incorporated that into the beta body. So GA is a glutaminic acid. And as you can see, there's up to 3 glutamic acids in these structures. And they are designed as peptides that sit either on the bottom side or on the top side of the fibril that grows. So this is what it looks like. You can either have them on the top side or on the bottom side. And then some of them are just L amino acids, some of them are all D amino acids. So we have a variety of different peptides here. And now some of them work fantastically, some of them work less. So if we combine the different peptides here, this was the former experiments, and you can see there's one combination, 2 and 3 here, this one and this one. So that's the old one, and this is one of the new ones with GA. That works pretty good in inhibiting the fibrillation. They don't fibrillate by themselves. Okay. I don't know what goes on here. That was it. Yeah, so we looked at self-fibrillation, and when we mix them, there's no problem, no fibrillation at all by themselves, and we still have very good fibrillation of the beta 1-42. So then when we look at the interaction with beta 1-42 with the different combinations of peptides, we found that this 3 and 5 actually form a very, very strong interaction that completely inhibits fibrillation of 1-42. It was designed for the crystal structure of 1-42, so that was also expected. It also works for 1-40, another structure, but that doesn't really crystallize as well in that particular structure. So here the effect is less than it is for the 1-42. But we can completely abolish this fibrillation. So we have 4 kind of click reactions. We have this CuAAC reaction, we have the spark reaction, we have a TCO reaction, and we have this ENIAC reaction that I haven't been talking about, and I won't talk about that today. So we are back in Stockholm, and here you see the dress, which is the click dress. It was made by the artist school in Stockholm. And it's fantastic, except for the fact that they are not chemists, so they have made 6-membered ring instead of 5-membered rings. And another thing you will notice is that it's actually my wife who's wearing it. She is very proactive, and she went to the design school and said, "Can you make this for me for 24 hours from now?" And she's carrying this on to the ceremonial dinner. And here is then what happens when your wife dress up like that. She was selected by the Prince of Sweden as a lady of the table. So I was sitting down there, and she was sitting between Daniel and Philip. But next day we had a very intimate dinner up at the Royal Castle. In this intimate dinner, you have a lady at the table. And in this case, I got my revenge, because this is one of the most beautiful ladies in the world. This is Crown Princess Victoria. And you can see how happy I am, right? So the last thing I will talk about today is our attempt to cleave Alzheimer peptides with catalytic peptides that has metals bound to them. So we made these building blocks which can form carbenes in peptides. We treat it with strong base and add the metal, palladium usually, and then we look at, can we cleave something with that? So that worked actually fantastically. So we had this peptide that would line up with the beta extended structure for the Alzheimer and cleave it. Wow. So it worked very well. It actually worked so well that we could start reducing the amount of the cleavage peptide, and we reduced it and it still worked, and we reduced it again and it still worked, and then we took it out completely and it still worked. So what was going on was that we actually had discovered that these peptides, they are auto-hydrolytic. They are forming a cascade reaction that takes place after a while. So first you have formation of these active agglomerates, and then they start hydrolyzing themselves very fast once it starts. So that's what I want to show you here. We have all the different substrates from the Alzheimer peptides. And we can then see we have very specific cleavage here. So these 4 bonds are cleaved. Usually it's the F4 and the F0 to start with, and then we have some exoprotease activity towards the center. We made a lot of different peptides to test this out. There's 14 to 24, 14 to 24, 12 to 25, 12 to 25, 1 to 28, 1 to 40, and 1 to 42. All of these, we did this kind of investigation on. So here I just want you to focus on this one, which shows you the starting material. And then after 9 days, we still have a lot of starting material. 10 days, 12 days, something happening, right? 17 days, and then it's all gone. So this is like an autocatalytic cascade reaction that happens at some point. We get the right conglomerate that has the right proteolytic activity, and then it goes very rapidly. So that's kind of interesting because Alzheimer's is, of course, something we would like to get rid of, these peptides. And there could be a real biological mechanism in this regulation that involves this kind of hydrolysis. We don't know yet. We could follow it kinetically and see how things build up and disappear again. And they end up with these peptides finishing here at the glutamine and here at the phenylalanine. The green part there is the nucleation site for the fibrillation. We also follow this by atom force microscopy. And you can see initially in this process, we form these donut-shaped structures. They are composed of 3 domains with some thin slices in between. And our thought is that maybe it's between these domains that you form these proteolytically active residues. We can also do a cross-seeding. So we can take an active substance from one container and put it in here to 1 to 40. So here see 1 to 40, completely stable if we don't do anything. And if we cross-seed with one of the proteolytically active ones, you can see it completely disappears. So that's just a tiny amount of catalytic material can start this process. So we have a proteolysis of the fibrils happening at the same time as we have the formation of the fibrils. So there's a balance of some kind in this whole process, leading eventually either to solidified fibrils here or to debris that is washed out from the brain during our deep sleep at night. And now it would be nice if we could make that change. The escape button, yes. I'll escape. And then we'll try again. We'll escape again. Yeah, it does change here, but not there.
Follow your same
yes.
Same schedule.
So
same procedure.
Yes. Escape.
Or what do we do?
Yeah.
We close it and open again, right?
Yeah, but we closed.
Yes.
Just a second. It should be over there. If we close this now. Yes, and now we turn it on again. Sorry about that. It's sort of ruining the whole thing. There we go. And now we go to the next slide. And this is showing you the donuts that we actually observe and a modeling of those donuts. So before we get the fibrillation in long fibrils, we most likely get these kind of donut-shaped structures that we also see in these nano images here from atom force microscopy. Our thought is that we make something that looks like that, composed of different fragments with a proteolytic activity. So this is a hypothesis. We haven't shown that we actually have this agglomerate there. It looks very much like a protease, though. And here we are. At the end, I would like to say that think we have a lot of new insight into the Alzheimer peptides. It seems that we can prevent fibrillation. Whether that's a good thing, we don't know, because it could be that these proteolytically active parts that we get by defibrillation could be toxic to the brain itself, just like the proteases are toxic to the host organisms. So Alzheimer peptides, they self-organize into these globular modules that can be proteolytically active. There's a long lag phase in this process, so that fits with the fact that with precipitation you get the fibrils. Just small amount of dissolved protein can eventually make shorter and shorter fragments and get this autocatalysis to go later on in the development of the disease. And seeding can allow us to cleave this at will for even the longer peptides. So that's conclusion there. And then the conclusion here, all the people that have worked with click chemistry over the time in my laboratory are listed here. Klaus Bock, one of my mentors, I have the title, A Leap from Shoulders of Giants. He is one of them, but there are so many I won't be able to list them today. In NMR and structural biology, in peptide synthesis, in polymers, and so on. And then there's my family, Fedra, who is here today, Adjani, my son, Anna, and Sandra, who has all been very patient with me because I spend a lot of time in the lab, I must say that. And then they've also been very good in discussing the science, all of them. My son is today actually studying chemistry, so that's very nice. And thank you for your attention and for being here, so many of you.
Thank you very much. Thank you very much. I think Professor Meldal has really shown us the power of fundamental research, the development of new synthetic tools, and their unprecedented impacts on tackling the real-world challenges. So now the floor is open for discussion. Do we have any questions from the audience?
Yes. I should say there's no such thing as a stupid questions. They are all good questions.
So, Morten, this is, I've heard you talk before, and, you never, it's just not the chemistry, it's your personality that shines through. And I'm sure every student in this room now want to do more science just to be like you, because you're the fun scientist. Question on, and actually the last part, the Alzheimer's part. This is really interesting looking into the mechanism, but then you still have the issue of delivering the stuff, right? So I don't know if you have given an idea on how this kind of molecular recognition can work to deliver this material where it needs to be. Thank you.
I was working with a co-worker at another institute, a health institute in Copenhagen, on making nano containers, contain our molecules, and had the ability to cross a blood-brain barrier, and had the ability to sit on plaques. So this would be a very good approach, but unfortunately, he stopped and went to the US or somewhere, and I haven't had contact with him since.
Maybe you need to look for a new collaborator. Like me. Was I very okay. Very subtle.
Great talk. So my question is, so the click chemistry is a sort of scratch on the surface to delve into the biology from chemistry, right? I mean, biology has very, very fixed, rigid guidelines of peptide chemistry, sequences, enzymes. They're all well established and factories of chemicals. But then you come in with a copper, you come in with something completely foreign to the biology, well, not too foreign, but somewhat foreign. So can we expect like synthetic polymers, polyamides, sort of making way into the brain and sort of replacing some of the therapeutic systems, like targeting reactive oxygen species or whatnot?
Yes, I'm sure that can happen. There are peptides that actually cross the blood-brain barrier very readily. And these are usually unmethylated because the problem is the hydrogen bonding network does not allow you to transverse usually. Some odd cases they do. You can even have antibodies transported across the blood-brain barrier. So to find out what happens there is a major issue. And how to actually utilize the natural mechanisms for transport.
I'm talking about the polymer chemistry. Like there's a
Yeah.
whole body of non-biological polymers.
Yes,
out there.
and they can be used for biological purposes.
Yeah.
I know a guy, so Professor Mark Bradley in Edinburgh University. He actually did 2,000 different polymers with different components in a combinatorial fashion. And he then grew stem cells, and he could actually differentiate stem cells within this polymer network. He could have the stem cells growing, eventually eating the polymer, and then becoming the tissue with the shape of that polymer. He's done some very interesting thing with that. And so that means a polyamide of completely unnatural structures are interacting with the biological system and is controlling the differentiation of the cell.
Thank you.
Because we are under a tight schedule, let's allow one further question. Yeah, please.
Okay, thank you very much for such an amazing
Is there a
talk.
microphone?
Does it work? Oh, perfect. Thank you so much for an amazing talk. I have one simple question. So you have that amazing system with your beads. What is the temperature limit of it?
The temperature that we use with these beads, usually these bioassays are carried out at room temperature up to 37, depending on what it is. But we can take these beads up to 150 degrees without degradation of the polymer network.
Okay, thank you so much.
So we do organic reactions. For example, I have a ring closure reaction that takes 130 degrees and so on.
Okay.
Okay, then please join me thanking Professor Morten Meldal again for the wonderful presentation.